Okay, folks, so we don't just stop monitoring the drug once it is FDA approved. The drug enters what is called phase 4, and this is really ongoing. Doctors will continue to look for side effects of this drug. As the drug is used by more and more and more people, we will expect side effects to pop up. And sometimes when we use the drug on enough people, a rare but very serious side effect might be visible. For example, there was a drug that had met FDA approval called rosiglitazone, and it was used. But once it was used by many, many thousands of people, it was withdrawn from most markets, not the United States market. Why? Well, this drug increased insulin sensitivity, which was an unexpected side effect, and increased the risk for heart attack and death in a way that many thought was unacceptable. So drugs, once FDA approved, can possibly be removed from the market. And so now, this has been your overview of clinical development of drugs from phase 1, 2, 3, FDA approval, and 4. And before we leave, what I wanted us to do just a little bit here, folks, is talk about the pros and cons of clinical trials. So the pros of clinical trials, right? They are rigorous and blinded. What does this mean? This means oftentimes the scientists performing the clinical trial don't know what drugs the patients are being given. And this is incredibly important. We need these trials to be blinded, because if the scientist or if the physician knows what treatment a patient is given, they will likely act in a way that is biased. So maybe the patient that's receiving the real drug gets better care than the patient receiving not the drug in development. We need these patients to receive the same treatment from physicians. Similarly, what if the scientists know which data points received what drug? They might analyze the data in a way that is biased to tell a better story, for example. So these clinical trials are organized before they start in such a way so that bias is at a minimum. Pros of clinical trials also, ideally, you end up identifying drugs that work in humans. Clinical trials are done on humans, folks, right? Preclinical work is not. If we want to be developing drugs that help us beat a variety of diseases, we need clinical trials to do so. We can't just be randomly approving drugs without testing them in humans. Additionally, clinical trials are good because we have kind of outside monitoring through the FDA. It's good to have that peer review to ensure that the data is real. Now, there's not just good things about clinical trials. There are some cons of clinical trials. Well, they are extremely costly. It is estimated that a single clinical trial for one drug is about $1 billion. And so that is a lot of money to spend on any one drug. Additionally, clinical trials usually take a long time, and this can be seen both as a pro and a con, in my opinion. I think it's a good thing that clinical trials take time so that we are making sure that we're gathering good data. We don't want to be jumping to conclusions without sufficient data to support approval of a drug. However, this means that it can take longer than is absolutely necessary for a drug to reach the market. However, we did see, for example, during the COVID-19 pandemic, that sometimes the approval process can be expedited in extreme circumstances, such as a global pandemic, where the vaccines for COVID-19 were kind of pushed through this process faster than they normally would have been. An additional con of clinical trials is that almost 90% of drugs will fail clinical trial and not make it to FDA approval. So this $1 billion being spent per drug, largely, you can think about it kind of like this, it goes to waste. Not necessarily wasted, because it's good that drugs that don't work or aren't safe aren't making it to people, right? But sadly, a fact is that some clinical trials fail because they weren't designed appropriately. If the right controls aren't set in place, if the hypothesis isn't developed in a way that is specific enough or rigorous enough, a clinical trial can fail, just due to its design, not necessarily because the drug doesn't work. And so, I'll give you kind of this graph as kind of an ending of our time today. This data is from a paper showing you what kind of the likelihood is for drugs to make their way from phase 1 all the way through the phases of clinical trials to FDA approval. You can see that taken together, on average, about 90% of clinical trials fail because only about 10% are successful. But the success of drugs making their way through clinical trials depends on what disease they are being developed for. You can see, for example, that cancer treatments are some of the least successful drugs going through clinical trials compared to drugs for blood disorders. Those have a more than 25% success rate. So, of course, the disease you're developing drugs for matters as well. And that wraps up our discussion of clinical trials. We worked our way through the 4 phases of clinical trial. Phase 1 is safety. Phase 2, efficacy. Phase 3 is efficacy and safety in a much larger population. And then phase 4 is constant monitoring after approval to make sure no very severe side effects pop up. And with that, I'll see you next time in class.